The human lipidome contains a persistent kinetic trap. At the cellular scale, the biological half-life of the most abundant dietary fat in the modern diet is ticking down from approximately 680 days. That fat is linoleic acid, a diunsaturated omega-6 essential fatty acid. Because mammalian cells lack the desaturase enzymes necessary to create double bonds beyond the carbon-9 position, every molecule of linoleic acid in your tissue is derived from dietary intake. Once esterified into subcutaneous adipose tissue, linoleic acid remains sequestered. Its metabolic half-life spans roughly 680 days, representing a multi-year biological sentence. This timeline identifies the kinetic disparity between dietary fats. On the top, saturated fats like palmitic acid, which the body synthesizes on demand, exhibit rapid metabolic turnover and are routed towards cellular energy requirements. Below, linoleic acid occupies a 680-day retention curve, resisting rapid clearance. This shift reflects a structural change in human physical composition. By tracking subcutaneous adipose tissue, we find a historical baseline of 9.1% linoleic acid in 1959. By 2008, that concentration reached 21.5%, a 136% population-level increase in tissue saturation. This accumulation correlates with the mass industrialization of the food supply, specifically the scaling of commercial seed oil manufacturing. Seed oil consumption triggers a long-term biophysical restructuring of the human lipidome, creating an unstable foundation for systemic cellular failure. The consequences of this restructuring are most visible within the inner mitochondrial membrane, the site of a unique dimeric phospholipid called cardiolipin. Mature cardiolipin requires 4 linoleal acyl chains, derived from linoleic acid, to achieve its conical shape. This structure induces the membrane curvature required to fold mitochondrial cristae. In physiological concentrations, this molecule stabilizes the electron transport chain supercomplexes, maintaining the structural integrity needed for ATP synthesis. Pathology begins when excessive linoleic acid saturation meets cellular oxidative stress. The bis-allylic carbons are vulnerable to hydrogen abstraction from reactive oxygen species. When oxidized, they trigger a lethal interaction. Cytochrome C anchors itself to the compromised lipid chains, converting into a cardiolipin-specific peroxidase. Using local hydrogen peroxide as a substrate, this complex catalyzes the peroxidation of the remaining linoleic side chains embedded in the mitochondrial wall. The oxidation of these structural anchors dissolves the lipid rafts holding the electron transport chain together, halting oxidative phosphorylation. This induced lipid peroxide accumulation triggers the opening of the mitochondrial permeability transition pore, causing the leakage of mitochondrial DNA into the cytoplasm. By saturating the inner membrane with fragile polyunsaturated fats, the cell weaponizes its own respiratory proteins to catalyze the peroxidation of mitochondrial walls, dismantling the energy grid from the inside out. This lipid peroxidation extends to the brain's angioarchitecture. The blood-brain barrier relies on microvascular endothelial cells to separate the central nervous system from circulating blood. The paracellular space between these cells is sealed by tight junction proteins, specifically Claudin-5 and a scaffolding protein known as ZO1. These function as taut cables. When the linoleic acid stored in these membranes undergoes peroxidation, it generates a reactive aldehyde called 4-hydroxy-2-nonenal, or 4-HNE. 4-HNE acts as a reactive electrophile, targeting cysteine and histidine residues forming the backbone of endothelial proteins. Colliding with Claudin-5 and ZO1, it forms covalent Michael adducts, inducing physical distortion. These modifications cause the junctional complexes to depolymerize, detaching and breaking apart under tension. This structural disassembly results in a drop in transendothelial electrical resistance, eliminating the barrier selectivity. Without these tight junctions, neurotoxic plasma components, albumin, and circulating bacterial lipopolysaccharides infiltrate the brain parenchyma through the resulting paracellular leak. Simultaneously, 4-HNE depletes intracellular glutathione pools. Without this primary antioxidant defense, endothelial cells undergo iron-dependent ferroptosis, creating structural voids in the barrier. Under oxidative stress, bioaccumulated linoleic acid transforms into molecular shrapnel, severing the brain's physical immunological cables and inducing neurovascular inflammation. This pathological reality faces a medical paradox. Replacing saturated fats with linoleic acid lowers total circulating LDL cholesterol. We can analyze this discrepancy through the lens of recent medical debates among experts evaluating cardiovascular risk and lipid clearance. The mechanism is documented. Substituting saturated fats with polyunsaturated fats upregulates hepatic LDL receptors. This clears a large volume of ApoB-containing particles from circulating blood, reducing the probability of physical particle retention in the arterial wall. However, this reduction in particle count ignores the particle's chemical vulnerability. The remaining LDL particles undergo core remodeling. One type is packed with stable saturated lipids, the other with fragile linoleic acid. Under oxidative stress, these PUFA-rich particles undergo rapid lipid peroxidation. Free radicals pierce the double bonds, forcing a transition into an oxidized state, ox-LDL. Once oxidized, the apolipoprotein B ligand on the particle surface is chemically altered. It is no longer recognized by hepatic LDL receptors, preventing standard clearance. Instead, these unrecognized particles are scavenged by macrophages in the subendothelial space. This scavenging mechanism drives the formation of foam cells, triggering endothelial inflammation and accelerating the progression of arterial plaque. Replacing saturated fats with seed oils compromises particle integrity, trading a statistical reduction in cholesterol for a highly combustible vascular system. While bioaccumulation is persistent, the mathematical half-life means this tissue saturation is reversible through directed lipid remodeling. Protocol 1 requires halting all exogenous loading. To stop the influx of oxidized linoleic acid metabolites, the dietary supply must be restricted. This requires the elimination of heated industrial seed oils, preventing the direct exposure of the vascular endothelium to reactive aldehydes like 4-HNE. Protocol 2 initiates the omega-3 index reset. Increasing the intake of EPA and DHA competitively displaces linoleic acid from cellular phospholipid bilayers, reducing the substrate available for peroxidation. Reaching omega-3 saturation restores the transcription of occlutin, allowing the blood-brain barrier to repair its tight junctions. Protocol 3 is the transition of background energy towards stable, low-oxidation fats. Historically dominant dietary fats, specifically those rich in stearic and palmitic acids, are resistant to free radical autooxidation. Shifting the dietary lipid ratio systematically purges 4-HNE precursors from adipose storage and resecures systemic membrane integrity. Biological degradation is cumulative, operating on a kinetic timeline that spans years. In the hierarchy of health optimization, long-term cellular destiny is dictated by the physical integrity of your membranes. The 680-day biological sentence is ticking. The time required for clearance is fixed. The metabolic override and clearance process begins today. Secure your biological future. Subscribe to the archive for Audit 003. The forensic audit of systemic seed oil contamination is complete, and the file is closed.